My topic of today's title is Screening Techniques for the Identification of Bioactive Compounds in Natural Products. The purpose of this topic is, as we all do a research in two types. One is target-based research. Another one is organ research, means nothing but classical pharmacological research. What today, which I am going to explain you various techniques, these techniques which are target-based approach techniques. Target-based approach technique. I hope you maybe heard yesterday's class by Kavimani sir as well as Srinivasulu. They have given the various approaches in the drug discovery. One is classical approach, another one is target-based approach. Whichever the experiment models or whichever the techniques which I am explaining you here in order to identify the bioactive compounds in natural products, all are target-based approach. Nothing but here there is a instrument called as pharmaceutical analysis instrument, and from pharmacology there is a target protein or target tissue or target cell membrane. These two are mixing together and they are seeing what is the binding capacity, and they are analyzing the particular active constituents. Let's have discussion one after the other. Content, I already told. The cell culture based assay is the one experimental model. What is the basic principle involved in each of the experimental methods? And one example, some of the plants they have used and they have identified the particular bioactive components from the particular plant. We will discuss each of the experimental models one after the other, followed by what are these limitations of these biological screening methods, what are the future perspectives of these biological screening methods, and followed by I am going to conclude my lecture. As I told you, sorry. Okay. As I told you, these natural products have a long history of clinical use and as well as a rich source of bioactive components. Having a rich bioactive components, development of tools and techniques are also at the same time very, very important in order to analyze the natural bioactive compounds from the natural products. Why? Because we have to ensure the quality and discovery of new drugs is very, very essential. That at the same time, these are complex mixtures and having hundreds of chemical constituents. That is the reason not a active single constituent will be effective. It may be multiple active constituents may be responsible for the efficacy. In order to have a particular efficacy and have a responsible pharmacological action, we should have a proper method or proper tool in order to identify the particular bioactive compounds. If you have a tool and if you have a techniques, definitely that will help you to discover a new drugs that is highly demand at presently what actually the research is going on. This is a just brief. I wanted to make you to know various milestones in the discovery of natural products. Just I am taking since from morphine in 1803, the morphine was isolated and it was the first drug became a pure drug as a natural derived medicine, and followed by in 1822, 1850, quinine, colchicine, codeine, papaverine, these are the drugs they have discovered. And 1929, Alexander Fleming, they discovered the penicillin, followed by 1952, streptomycin, another antibiotic they have discovered as a natural bioresources. And 1959, citrabirin, it is an anti-cancer drug which they have discovered from the plant source. Then 1990, various pharmaceutical companies, they started working on using the computational chemistry and high throughput screening. They have started working on various plants. And you know very well, 2015, one of the scientists, they got Nobel Award for the discovery of artemisinin as an anti-malarial drug. These are just a milestone of natural products research discovery. Now, I'm going to explain usual experimental methods or techniques to identify the bioactive compounds. I am wanted to you to know, as yesterday, Dr. S. Kavimani sir and Dr. Srinivasal sir, they told is classical approach, another one is target-based approach. Nothing but a classical method, what it involves. Hello, I am audible. I am audible to you all.
Yes, sir. Yes, sir. You are audible, sir. Carry on, sir.
Okay. The classical method for screening of bioactive components. What is the basic principle involved in classical method, and what is the basic principle involved in target-based or modern technology, which I am going to explain you today is modern technologies which they are useful. In what is this in classical method which they are doing? As we know all, as I am a pharmacologist, I am going to take a plant and I am going to extract that particular plant, and then I am going to determine some of the pharmacological activities. Once it is showing some pharmacological activity, then I am going for pharmacokinetic studies and some toxicological studies. Then I am going to know about safety of the particular compound. This is the basic classical pharmacological approach in order to identify the bioactive compounds from the natural. But high throughput screening assay, this is, you know very well, all of you, there are number of targets and number of different types of cells and number of compounds which can be screened simultaneously, and you will get number of results and number of pharmacological actions which we can be analyzed by using HTS, means high throughput screening. This all you very well you know. But this classical approach, what is the problem is, it is also a very effective method. I am not telling it is classical approach is not a good effect. It is a good method and it is effective, but the thing is time consuming and it is a laborious, this classical method. Now I am going to tell you, see here, what we are using is forward pharmacological approach. By using, we are extracting the components and we are injecting to the animal, and from that animal we are going to be able to identify the target identification, that is called as pharmacological approach. Now this is called as reverse pharmacological approach, which I am going to, whatever the today's techniques which I am going to explain to you, this is my approach. This is called as reverse pharmacology approach. Nothing but first we will identify the target. Target is nothing but, you remember, a cell protein or a enzyme or a DNA or a protein of the particular enzyme. Say for example of aldose reductase is the enzyme. That enzyme which I am going to take first. Then I will going to incubate that enzyme with the particular test extract or particular compound. Then I am going to see how much the drug is, how much that extract is going to bind to that particular membrane or particular enzyme or particular protein. Then after that I am going to separate, after whichever the compounds which are going to bind to that a particular enzyme, that I am going to take and I am going to separate it, and I am going to see, oh, that these are the compounds are responsible for so and so particular enzyme inhibition. That is my today's explanation, that is called as reverse pharmacology approach. Reverse pharmacology approach. This is forward pharmacological approach. It is a time consuming and it is a laborious process, but reverse pharmacological approach is very fast and high accuracy. You will be going to get, and you will get mechanism of action of a drug also you will going to get from this reverse pharmacological approach, or nothing but target based approach. I hope. Listen to me now here. The modern pharmacological studies. What this modern pharmacological studies? Nothing but target based approach. What they demonstrate? They demonstrate the drug action is determined by its ability to interact with, as I told you, ability to interact with a target protein or a cytosolic protein or a transmembrane protein. Nothing but DNA, nothing but enzyme or nothing but receptor. They are whatever the extract or whatever the chemical compound which they are going to demonstrate by using a cytosolic membrane or a transmembrane protein or by using DNA or by using enzymes. This is a modern pharmacological approach. That's what a bioassay guided isolation and screening process can increase the drug discovery process by identification with the medical relevance. That is the reason several biological screening methods, you see, several biological screening methods have been developed for multiple biological targets. There are many methods are there, which I am explaining you one after the other now after coming slides. These screening methods, they involves the target proteins called as DNA, protein molecule, enzymes, or receptors, which I am going to show you in the table 1 and figure 1. You can see these are the extracts, ginkgo biloba. There are many extracts are there, and cells, mesenchymal cells, splenocyte cells, human umbilical mammalian cells, artificial membrane, XO, you see, xanthine oxidase is the enzyme. This is acetylcholine esterase is the enzyme. COX-2 is the enzyme. And the methods which I am explaining you one after the other, these are the cell with natural product extract incubated with, and HPLC MS is the instrument which they are used. Like this, many microdialysis technique is there, liposome chromatography column is there, like this many ultrafiltration is there, one more technology. These are the techniques which they are used in order to identify the bioactive components by using the cell targets. These are the targets, and they are able to identify the bioactive components and as well as mechanism of action of that particular active component also. Remember, see, this is the table 1, then the table figure 1. See, these are the cell is a one target. Biomembrane or cell membrane, nothing but liposome is also a cell membrane. And target protein molecules, say for example a protein, DNA, enzyme, receptor, these are all called as targets. Then what they will do, these targets and extracts, plant extract, this is the plant extract with having a various active constituents, and this is the target. These two are going to mix active components and combined with the targets, that is extract targets and active components, they're combined together, and they will be, say for example, of the targets, these active constituents are going to bound to the target and unbound compounds are washed away. Then bound compounds are separated by using some chemicals or digesting, then separating these chemicals are active constituents, and waste products will be removed, and they are going to analyze this by using HPLC, LCMS, some other instruments. I hope you can see here, these are the technologies. One is cell culture, another one is equilibrium analysis, and another one is microdialysis, ultrafiltration technology, chromatographic method, and another one is target molecule immobilization method. All these methods, the basic principle is this. This is called as target-based approach. Whichever the models which I am explaining you, this is the basic principle involved in all of the experimental models which I am explaining to you one after the other. Target may be cells, or maybe biomembranes, or maybe target molecules like DNA. Receptor. Yes, my dear colleagues, friends. Hello.
Yes, sir. Sir, you are audible, sir.
Okay,
it's clear, sir. No issues.
Just I wanted to confirm, sir.
Yes, fine, sir. Fine. No problem, sir.
Okay. That's what. As I told you with this diagram, the principle of modern pharmacological studies, is nothing but a target-based approach. What is the basic principle involved in this? Here it's very simple. A biological targets are incubated with natural product extract. Once they incubated, the biological target and natural products, they are incubating. Then whatever the potential bioactive compounds present in that extract will going to bind to the target molecule. Target molecule is, don't get confused, target molecule it is either cell membrane or a protein or enzyme or DNA. Means potential bioactive components will going to bind to the target molecules. Then unbound compounds will be washed away by using some certain buffers. They will be washed away and they get away. Then the active compounds isolated from the targets. Whatever the bound compounds are there, those compounds we have to isolate it. Then isolated after that, we have to analyze by using suitable chromatographic techniques or some other techniques. You will be able to, by using other detectors, we will be able to identify the active constituents which is there in that particular extract. And also at the same time, using bioinformatics technology, we can able to apply to get some structure and activity of that particular. That's what this technology which will enable us to identification of potentially bioactive compounds and also provides an information regarding their mechanism of action at molecular level and cellular level. Remember, this classical approach is at the end we will get the mechanism of action by doing so many chits of time and so much of activity. But here you will get immediately after the application of that what actually a mechanism of that particular active extract. That is modern. One thing which I wanted to share with you all, see TCM, you read what is this TCM? TCM is nothing but traditional Chinese medicine. Why am I stressing on you TCM, traditional Chinese medicine? See, China, whichever the models which I am explaining you, I referred many of the research papers, models which they have been used for their traditional medicines in order to identify the active compounds from the natural products. This is their standard setup methodology in order to identify the active components from the natural products. They have standard setups. You can see, if you go for in vivo, these are the setups. If you go for the in vitro, these are the setups. And you see biochromatography, they are using molecular biochromatography, cell membrane chromatography, artificial biochromatography. Like this, so many models, methods they are using, and whichever the research papers which have been come across in order to prepare my slides, all their research is using by this technology. Most of the researchers are from China. That much they are good in using this technology in order to identify the active constant as well as mechanism of action by using modern pharmacological techniques. As we, as a pharmacy students, as we are in the pharmacology, we have to approach this way, which I am asking you all to know about this and try to do in your lab, which I am going to explain you which are the models now. That's what the models which I am going to explain to you today, one after the other, another 30 minutes. One is cell culture based assay, equilibrium dialysis assay, microdialysis, ultrafiltration, chromatographic, and ligand fishing based immobilization of the target molecules. Let's have, no need to waste the time, one after the other we will discuss. What is this? This is important and plays an important role in cleaning up active components from the natural products. What they do in these screening procedures, cells, particular cells, say for example a liver cell, or it may be hepatocytes, or it may be kidney cells, any cells which they are mixed with the extract of the natural products, then the natural products are going to bind to the cells, and which are not bound, that is nothing but unbound compounds are washed by using with the buffers and they remove. Then the cells and selectively bound components are then digested with some chemical components, then analyzing the components by using chromatographic techniques is the method. Depending upon the culture method, there are two types of method. One is classical cell culture method, another one is hollow fiber cell culture method, which they are using. This is one of the researcher, Yuan et al. He has studied, isolated the candidates bioactive compounds in Panax ginseng. Panax ginseng is the compound plant. They have extracted the compound for two neurodegenerative disorders by using a technology cell culture based assay. They have taken as a cell line SH, they have cell taken and they have incubated and allow the interactions of extract and the cell lines, and then after that they centrifuge and precipitate, washed with the phosphate buffer solution to remove the unbound compounds. Then after that, bound compounds, they are going to denature with the 40% acetic acid extract by ultrasonication extraction by methanol, and followed by another centrifugation step. Then they are separating the active components which are bound to the cells, and they are going to analyze it by using high performance liquid chromatography coupled with quadrupole time flight mass spectrometry. And they have identified potential active components from ginseng in this study. You see, this is, I hope you can see, these are the active components of the ginseng extract, and this is the target cell. They are incubating with the cell culture flask. See, now the brown color, I think, brown color is the active components which are bound to this cell, and this yellow color, they are not bound to this. And after washing, the yellow color components are washed away, and whatever the bound components with the cells are there here. Then after that centrifugation and sonication, they are digesting and they are separating from the cells. These are the real active components, and they are going to extract it and they are going to analyze by using LCMS. Understand? This is the actual cell culture based assay. No need of using any animals over here. Only cell lines is the target agent. Yes? I hope people are listening to me.
Yes, sir. Yes, sir.
Yes, sir.
Yes, thank you. Now, next one more is there. Hollow fiber cell culture based screening system. Hollow fiber cell culture based screening system. This is also one more widely used model. Simple, relatively speed, and have a high enrichment. You can do it, and solvent consumption is very less and low cost. Some researchers, they have done using Rhizoma curcuma longa and Rhizoma curcuma. How they are using this method? In these studies, what they do, hollow fiber cell culture method, they are taking number of living cells. They are injected into the fiber lumen fiber. There is a fiber, I will show you in the diagram. Living cells, they are going to insert into the fiber tube with the help of syringe. Then that hollow fiber is going to bend like a U-type shoe, and inserted into the natural product extract culture flask, and they will allowed to bind to the fish active compound. The compound, fish active compound, will be going to bind to that fibers. Then the natural product extract was stirred. They are going to stir at 37 degree. Then the hollow fiber will be removed from the natural product extract solution, and seals of the both ends were removed, and hollow fiber cells with target analyte was dissociated with methanol centrifugation. Then the supernatant was subjected to HPLC analysis. You see here, they are taking the cells with a syringe, and needle is fixing. Then after fixing the needle, these cells, they are going to inject into the hollow fibers, and after that that U-tube are going to place it into a, you see, hollow fiber tube is there inside in this culture flask. Here is also you can see. This is called as U-tube. This they are going to place it into a culture in order to observe the plant active constituent into the fiber cell. Now you can see one more diagram that may be difficult to you to understand. See, this is the plant. Think of we having a extract and cells and having a particular target. And this is the hollow fiber cells, seeded cells, and this is the hollow fiber cells filled with targets. Either you can see this also, or either you can see this also. Both are same. They are going to put it in a culture flask, and they are going to stir this vial with the help of magnetic stirrer. Then after that, you see the compounds, the target as well as bioactive components are joined together. Then after that dissociation, then the compounds which are not binding, though they are going to eliminate, and the compounds which are bind to that fiber will be there here, and they are separating these active components. Then after to you see here the cells and as a targets, and they are mixed with the components isolated after that by digestion and dissociation. These are the compounds. By you will come to know structure also. You will come to know what is the mechanism of action of the particular compound also by using hollow fiber cell technology. What is the limitation of this assay? The biggest drawback is cell gradient. During the screening process, the cells have a different gravities and the concentration may be vary. At the same time, overall, the cell culture based assay is requires long time for incubation time, and it may be difficult for high throughput screening. This is about the cell culture based assay. Next one is equilibrium dialysis. This is also another techniques, screening techniques in order to identify the bioactive compounds from the natural products. What this technique says, this also techniques enables us to separate the bound and unbound natural product active constituents using a semi-permeable dialysis membrane. Who are very good in pharmaceutical analysis, they are all very well, they know what actually the dialysis principle. I am not going to be explain what actually the dialysis principle. That same principle which we are using here in order to isolate and in order to identify the bioactive components by using equilibrium dialysis. There is nothing but semi-permeable dialysis membrane is the membrane that will going to separate the bound and unbound natural product active constituents. The target molecules, active compounds, complexes are unable to penetrate the dialysis membrane. Means here very clear explanation is there. You can see when natural product extract and target molecules are mixed in a dialysis bag, any unbound compounds pass freely through the dialysis membrane and are eliminated. And the potential bioactive compounds, they cannot pass through the cell membrane. Then the dialysis membrane have a particular molecular weight cutoff. That is the reason that particular bound compounds are stay in the plasma membrane, that is called as semi-permeable membrane. Then after that, what they will do? See, the equilibrium dialysis, especially to isolate the active compounds with using HPLC or ultra performance liquid chromatography, then those compounds which are bound to this membrane, they are going to extract it and isolate it, and they are going to analyze by using HPLC or UPLC. Among this equilibrium dialysis, the one method widely used is liposome based equilibrium dialysis. Liposomes means nothing but lipids. Here you can see the liposome particles are there in a blue color, and free drugs are there which are going to bound here. They stay in the lipid in the dialysis tube, and which are not stay in this, they are coming out this. That is nothing but free unbound drugs. These are active constituent. They can P custodium. What they do? A mixture containing extract and liposome. I am audible to you. Hello, my dear colleagues, friends.
Yes, sir. Yes, sir. You are audible, sir.
Okay. Okay. A mixture containing extract and liposome, they are put it in a dialysis tube. See, a mixture containing extract and liposomes, they are put it in a dialysis chamber and having a tube. This is called as dialysis tube. And both the ends are tied. Both the ends are tied and placed in a glass tube containing a buffer for dialysis. After 24 hours of equilibration, what is called as interaction dialysate outside the tube was collected for HPLC analysis. At the same time, the blank dialysate also they are analyzing, and whatever the peaks in the interaction dialysate were significantly higher than the blank dialysate is indicated. There is a strong interactions of active constituent with the liposome, and they are isolating which are those active components from that particular plant. This is one of the easy techniques which we all can use in our laboratory by using dialysis chamber. And suppose if you do not have a LCMS or HPLC, there are many research labs are there, contract research labs are there. You can give the sample to them, then they will isolate it and they will give you, and you will get a good research publication in internationally reputed journals, which I am insisting you all to practice such kind of methods instead of going for animal studies and all. Just a by using bioinformatics and dark structure, they are able to identify the particular active constituent from this plant by using a technology called as equilibrium dialysis techniques. Now, next technique is microdialysis. This is a refinement of equilibrium dialysis. Only extra thing is here, a porous membrane is the extra which in this, and it is widely used for pharmacological and as well as physiological experiments, and it is a more attention towards the pharmaceutical as well as analytical chemistry. What is the basic principle with the one example which I am explaining you what actually this microdialysis, what they are doing in this screening method. They will take some particular target proteins, it may be enzyme or it may be cell membrane or it may be liposome, anything. They take the target proteins and incubate it with the extracts, and automatically once they're incubated with that, then they will going to place the probe, dialysis probe for a mixture of microdialysis. Once they are allowed for equilibrium, once the equilibrium reached, the microdialysate is collected for HPLC along with Can you hear me?
Sir, yes, sir. Share your slides once again, sir, please.
Suddenly it went. Sir, I think Zoom you are using is a purchased version, is it?
Yes, sir. Yes, sir.
We are not using purchase. You are using licensed version. I am not using licensed version. That is the reason they are going to block in between.
Okay, okay, sir. You can continue.
Sorry
now,
for
sir,
trouble.
please. It's okay, sir. It's
with one example of microdialysis, as we know all, they have taken as an enzyme. Here it is aldose reductase is one of the enzyme which is present in the human cells. It is very important and plays an important role in many of the pathological conditions of our diabetes mellitus. With this enzyme, we can able to prevent various diabetic complications such as neuropathy, retinopathy, nephropathy, cardiovascular complications. By using this enzyme as a target with the help of microdialysis techniques, a scientist called as Wang, et al., they developed a microdialysis system coupled with HPLC, high performance liquid chromatography, quadrupole time flight mass spectrometry. This is they have used for to identify the various target molecules from the Gardenia germinolides. From this, they were able to identify 23 potential active components for aldose reductase inhibitors activity. That's what you see. This is that. If you refer that research paper, you will be able to find. This is the research paper, rapid communication. This is the technology which they have been explained in various active constituents from Gardenia germinolides. Sorry to disturb in pronounce. Next technology which I am going to explain you, ultrafiltration. This is also one of the screening techniques in order to isolate and in order to identify the active components from the natural products research. Ultrafiltration is nothing but a semi-permeable membrane with a low molecular weight cutoff. They are giving a cutoff, a low molecular weight of compounds has to be get into this. This will this membrane will allows the liquids and solutes below a particular molecular weight will be passed through the membrane. It is a simple and reliability does not require any equilibrium procedure. Here we don't want any equilibrium procedure like 24 hours which we require in earlier models. That is the reason ultrafiltration is a more rapid screening method as compared to equilibrium dialysis or microdialysis. What they do in this with the diagrammatic I will explain to you. See here, this is the one enzyme, a target, and these are the a different active constant which may be the present in the extract, and both the membrane, semi-permeable membrane and extract, they are going to incubate it. Ultrafiltration system, the mixture of target and natural products occurs through a ultrafiltration membrane. Incubation, then fit it into a ultrafiltration with the membrane. Natural product active components specifically bound to the high molecular weight target molecules are retained on the membrane. You see here, which are the active components in the plant extract which are bind having a high molecular weight, they are retaining in the centrifugal tube, and some of them are come out from that. And after that dissociation, they are going to separate the target as well as active components. See, ultrafiltration, these are the compounds, they come out, but these are binding to the targets. And once they're binding target, then dissociation by using some technology, centrifugation and dissociation, then they are going to separate the targets and active constant. These are the active constant. Now they are going to analyze by using HPLC. See, this is one compound. This is yellow is another one compound. This is red is another compound. This is ultrafiltration technology. Same here also, extract of screening, and these are the screening target, and they are incubating and ultrafiltration, and the compounds are binding to the targets and wash. After that, unbound molecules go waste, and bound molecules will be stick onto this, and filters will release the ligand and these are the two ligands isolated from the particular plant extract. This is the basic principle involved in ultrafiltration technology. See here, mulberry leaves, they have used for activity, which I am not going to explain. See, this is the one of the research paper recently, I hope 2017. They have used xanthine oxidase inhibitors. You know very well what actually the xanthine oxidase enzyme inhibitors used for the treatment of hyperuricemia, gout and all. What we do in our pharmacology, we use a particular inducing agent in order to induce an hyperuricemia in an animal. Then we screen and then we administer, then we collect the blood sample, how much uric acid and all. No need of that. Now here, take a enzyme as a target and mix with the particular target. Target means xanthine oxidase enzyme is the target and extract, and they are going to use with the centrifugal ultrafiltration with the membrane, and they are separating and isolating the active components. What is the disadvantage of this method? Disadvantage is unbound compounds cannot be completely removed unless repeated manual washing is performed. That's what, in order to do this, in order to reduce a non-specific absorption of unfiltration membrane ligand, repeated washing of the cell membrane is very essential in this screening model. Next, I hope you know all chromatographic methods, and we all studied as a common subject in M-Pharma, pharmaceutical analysis. We know the basic principle, and at the same time, we know the uses of that particular chromatography or particular instrument. But the same uses which we are not applying as a pharmacologist, we have to apply. In that way, these are the experimental models which may help for all of us to understand and to identify the active constant. That one technique which I am explaining you, basic principle first I will explain. Here there are many techniques are there in bioaffinity chromatography. One we will discuss one after the other. What actually a bioaffinity chromatography? This is an all of you know the basic principle. Here bioactive compounds and immobilized targets. Bioactive compounds and immobilized targets. Bioactive compounds means from the natural products, and immobilized targets containing a stationary phase. Stationary phase is nothing but, as we know all, the column is filled with gel or silica. That is called as stationary phase, or nothing but a carrier. See, the targets combined with a carrier such as gel or a silica particles are used as a stationary phase. What happens? They selectively retain the potential active ingredient, specifically bind to that particular target, maybe cell membrane, or maybe protein, or maybe liposome. This is widely used experimental method in order to identify the bioactive components from the natural products. Very simple. Plant extract, they are going to inject into the column of an HPLC. That column is having a stationary phase containing a target. That is nothing but carrier and targets. See here, that diagram which you can see able to see. First, what they will do, the natural product extract is injected into a affinity column packed. Natural products extract, they are injected into an affinity column packed with target covered carrier under conditions. Target means cell membrane or particular protein or particular enzyme that allows the strong binding of interest compounds with the immobilized targets. The bound compounds are then dissociated from the target using a elution buffer. You know very well what is actually a technique of elution, which are then analyzed by using a suitable detector with the HPLC analysis. See, this is the carrier. Carrier means maybe silica gel and having a target. These are the cell, or maybe DNA, or maybe some particular enzyme, or maybe some particular protein, and these are the natural product extracts. See, once they are going to enter into this, and the bound compounds which are going to bound to these targets. These are the targets. Then after that, elution, with the help of elution buffer, they are going to remove bound compounds that is going to be act as a waste, and that bound compounds which are in the targets, they are going to isolate it by using some other techniques. Then these are the active constants which they are going to analyze by using HPLC. This is the basic principle in bioaffinity chromatography techniques. Today, the same bioaffinity chromatography, there are many different kinds of are there. One is cell membrane chromatography, another one is cell membrane affinity chromatography, another one is immobilized liposome chromatography, and biological molecular chromatography. Briefly, one after the other, I will explain to you. See, cell membrane chromatography. I am audible to you all?
Yes, sir.
Thank you, thank you, thank you. In cell membrane chromatography, the cell membrane fragment of a cell membrane, they are taking and they are going to absorb on a silica gel column. They are going to absorb in any of the cell fragment. Say, for example, if you know transmembrane, G protein coupled receptor is there. There is a 7 helical transmembrane is there. That's particular fragment of helical membrane they are going to take and they are going to absorb it into a silica gel, thus forming a membrane stationary phase. The stationary phase containing membrane. So some same biological activity as the same in the in vivo. Then the targets which are there, they are going to absorb, and the targets will absorb the active constant, and they are going to use HPLC analysis. This is a cell membrane chromatography. One example which I can give you, 2011, this is the research paper. This is again China peoples which they have published. Aconitum caramachili, they have used HPLC cell membrane chromatography in order to identify the active constituents from this technology. Next one is cell membrane affinity chromatography. Here also, cell membrane affinity chromatography, immobilization of a target of transmembrane protein onto a stationary phase. See, here, this is the transmembrane, TMPPRS. This is as a target and absorbed with the silica gel, acts as a stationary phase, and they are going to do next further process. What actually I told you already, basic principle, the same principle which will be applied for that, bound and removed by using other technology. Here you can see the best example, a brief sample and application buffer and chromatography having a targets and affinity of a ligand. Here these are the ligands, affinity, and these samples will go into bind to this. See, these are the plant extract or active components which are bind to the target, and non-retained samples are going out and retained compounds will be here. And after that, next step will be elution will be there, and elution is nothing but elution of buffer, change in the pH of our composition. Then bind compounds will be isolated and isocratic elution will be there. This is the way they are isolating the active components from the plant by using bioaffinity chromatography. Next one is immobilized liposome chromatography. Their transmembrane, here liposome they are taking. Liposome is nothing but a bilayer lipid layer. The most commonly used liposome screening model involves a steric immobilization of liposomes in a carrier. Then it is packed into a column. Then the compound of interest which will be added, and they are going to isolate. Why they are using this liposome? This is mimics the lipid layer by structure and fluidity of real biological membrane. That is the reason this technology is widely used. See, this is one of the research paper recently published in 2020. Development of an immobilized liposome chromatography method for the screening and characterizing the alpha glucose inhibitors. This was the research paper, the method they have used. We all can do by using such kind of methods. Now, another one method is there, biomembrane based bioaffinity chromatography. with the example which I am going to explain you. As we know all, one of the enzyme which is called as xanthine oxidase, which is going to generate a oxygen free radicals, and it may going to be responsible for the cause of hyperuricemia, gout, and chronic intestinal nephritis. Pharmacological inhibition of this xanthine oxidase enzyme may going to prevent various complications related to gout, especially naturally active compounds or plant-based compounds are widely used in order to prevent this hyperuricemia, or in order to inhibit the xanthine oxidase enzyme. One scientist, that is called as Peng et al. Again, these people are also China peoples. They have done with particular plant that is called as L-macronotide extract. See, this is the research paper they have used. Online coupling solid phase ligand fishing with high performance liquid chromatography, diode array detector, so with the spectrophotometry in order to identify the xanthine oxidase inhibitors. I'm not going to detail to explain, just basic principle, you know all very well. Another one is here, size exclusion chromatography. Here also, what they do, they recently developed a method, online size exclusion columns are coupled with column switching HPLC diode array detector. What they do here, screen a mixture of a biological molecules and a natural product extract. Any of the biological molecules which they are taking and they are incubating with the natural product extract, and directly without any complex procedure, they are going to inject into the chromatography, size exclusion chromatography, and it is having a very good advantage. You can see here the natural product extract with the biological membrane, and they are going to inject into a column, size exclusion column chromatography, and here binding and eluting waste and unbound compounds, and again they are using with the HPLC DAD. These are active compounds and these are inactive compounds, like that. Next one is ligand fishing based immobilization of the target molecules. Nothing but ligand fishing based target protein immobilization is also widely used method. What they do here is, the study demonstrates a protein immobilization on the magnetic beads that can be used for the ligand fishing. I will show you with the diagram, then only you will be easy to understand. Here you see, this is a protein coated magnetic bead, incubated with the test extract. This is the test extract, this is the plant, this is the extract, and magnetic coated beads incubating with that, and washing after that. Then some of the compounds which are bound to this, they will be stay and elute by using methanol. Then they are removing the which are bounding compounds, which are non-bound compounds, and they are using by elution of the buffer. Then they analyze the active components by using HPLC mass spectrometry. This is the research paper which they have used the plant Pururiae lobata flower identification of the active compounds by using this technology, which you all can refer this research paper further if you want. Again, limitation is natural products are complex, but here we are using as a single target. This is the reason many of the potential active compounds which they are binding, they may be not included in this techniques which we are using. All these, whichever the models which have been explained, the methods, maybe microdialysis, maybe bioscreening techniques by using bioaffinity chromatography or ultrafiltration. All these technologies, they are using single targets. But our active plant extracts are multi-components. We may going to lose some of the active components which may be helpful for the treatment also. That is the reason we may going to lose. That is one of the major limitation by using these techniques. Another disadvantage of these techniques is whatever the target proteins, whatever the molecules or whatever the enzymes which we are using is as a target, they have poor solubility. They are limit the use, reuse, and resulting a less efficiency and cost effectiveness. That is another disadvantage of using these techniques. And another major problem is that non-binding represents a major problem in a biological screening techniques. That is the reason non-specific absorption is primarily maybe due to because of non-active ingredients with the carriers and targets. Remember, because of non-specific binding is a major problem in this with the target molecule. We are going to lose some of the active constraints. And what are the futures perspectives in the biological screening techniques in order to identify the active constraints? The majority of whatever the experimental models I told you already is used for the screening is single target. Limits the amount of information that can be generated by using these techniques. There is a future scientist, as a future researcher, which we can develop a multi-target screening and analysis method so that we can use multiple active components for each medicinal herb can be screened and can be analyzed at the same time. That is the reason some of the now new technologies have come, a microfluidic chip technology, and some of the others they have reviewed biosensors is another emerging technology that can be integrated into a screening methods can be used. At the same time, one more, these techniques which have been used already, I told you, in order to identify the active components, same techniques which we can use for identify even the adverse drug reactions also. Why? Because we are going more natural products, and these potential natural products also may going to cause some of the toxic reactions. So that is the reason we can use these technique models in order to identify the adverse drug reactions or any toxic reactions from these particular active components. Why? Because the same techniques are same methodology, the same approach which we are using, which we can apply for even to identify the adverse drug reactions also. That I am wanted to tell you, stress onto here. Another one is, the problem is, if I am having a disease means I am going to take a tablet, and that particular tablet is going to enter into the blood, and blood is going to circulate all other parts of the body, then it will going to show us some action. Means drug absorbed into the blood and exert their bioactivities. But here we are using as a in vitro assays, bioactive components or metabolites of natural products in the plasma following oral administration is very advantage actually. But that is not taking place here. Everything is we are using with the chromatography and the mass spectrophotometry. In order to overcome as a future work, which all we can work as a biological screening, a target-based screening method that can be used directly to screen the potential active components from the directly taking from the biological sample. No need of particular target, which will going to improve the cost and as well as effectiveness of particular test compounds. This is a future which we all can think. Finally, I am going to conclude with my lecture having a following points. What and all till now that 40 minutes which I may be talk to you all, whatever the methods which I have been explained to you, these methods are highly efficient, stable and reliable biological screening methods in order to establish and analyze the active ingredients from the natural products. While focusing only in the primary and the identification of potential therapeutic components, similar system also can be designed in order to identify the screening of toxic compounds at the same time. That's what, at the same time, in future, faster and accurate mode of bioanalytical techniques are expected to develop for the screening of detection of active natural products from the compounds. Additionally, we all can give more attention should be paid in the development of biological screening systems. The system should be in such a way that should be capable of identifying the binding of natural components in a multiple targets. What are all the targets which we are explained here is single target. But the system of methods now we have to think and we have to work in such a way that multiple targets can be seen with the help of multiple components from the extract. That is interest which we can all work on to this. This is all about my today's class. Once again, I am going to thank you by saying, as we know all, COVID is the problem. Is be informed, be prepared, be smart, be safe, be ready to fight against this COVID-19. By saying this word, I am saying big thanks to Srinivasalu sir and Dr. Ishaq, who is constantly supported me, and especially thanks to principal sir and management for giving me an opportunity and being a patient of some of my problems which have been occurring in between. Really, I'm very happy to share what I thought to explain to you. I hope some of the peoples may going to ask any of the questions. Now we can open for the discussion. Thank you, sir. Thank you for giving me an opportunity.
Thank you, sir. Thank you so much. Really, your presentation is completely out of box. Since a couple of sessions we are going through, everybody are concentrating more on pharmacological things. As I belong to pharmaceutical analysis, I have keenly observing all the slides. It was really helpful for me also. Thank you so much, sir.
Really, these techniques which we have to use as a pharmacologist.
Yes, sir, definitely. These are the generalized techniques which will be generally used to any of the research area like pharmacology.
Any of the research area, but why in India, as a pharmacologist, we people are not using these things, directly going for extracting and injecting to the animal, and they could not able to publish any of the research paper. They are wasting.
Yes, sir.
At least, why? Because half of the work can be do it in our laboratory without any much expenditure. Yes, another half of the things which we can use, many research labs are there. HPLC, LCMS and all, we can give to the compounds to others to do that. Yes, why? Because whichever that have been come across by preparing these slides, all are China peoples which they have established their plants, active constituents very well by using these techniques. All advanced technologies they are using. That is the made me to give a lecture to our students or our faculty, our people should know. That is the reason I stressed on this. Thank you.
Definitely, sir. This is a need of the hour, what you had thought and presented the slides. Sir. Thank you so much.
Thank you.
Any of the participants, if you have... Okay. No, not at all, sir, not at all. If any of the participants, if you have anything to discuss with the speaker, you can kindly interact with him. Kindly unmute your mic.
Hello. Hi, this is Dr. Thippeswamy. This is Dr. Srinivas.
Yes, sir.
Yesterday's presenter.
Yes, sir. I know, sir.
Yeah, it's good that you presented these things. Actually, I did not touch upon the phenotypic drug discovery and technologies that can be used.
Yes, sir.
Because I totally wanted to touch upon target-based drug discovery that happens.
I understood, sir.
Good that you have these, whatever you have presented. I think most of the things, there is a common one terminology that in literature that is used normally. It is called as the beauty of some of the technologies what you have presented.
Yes, sir.
On one side, on one side you can do the extractions, bound versus unbound fractions, and
Yes.
extract you can extract the bound active ingredient and identify the ingredient.
Yes.
That is one aspect. The other aspect is for a particular active
And we
ingredient
can
of a
want to
particular
know some of the
extract.
mechanism of the particular compound.
Yeah. So the other way around,
Yes, sir. Yes, sir.
the other way around, the same technology can also be used to understand which particular target it interacts.
That's what, sir.
By incubating with various different kind of like the bead technology you have mentioned. So the
Sir.
thing is, what people do is they will the various targets they get attached. There are commercially available beads with various proteins. So
Yes,
they
sir.
will try to, they will, one particular compound they will incubate with several beads combined with, coated with several proteins. And
That's
they
what,
will
sir.
see which particular protein it is binding and then
Multi-components.
yeah, it's a dual technology, and the
Yes, sir.
process is called as something like what we call is target deconvolution, because you are trying to understand what is the target for the particular active ingredient. It is called
Yes, sir.
target deconvolution is a general terminology.
Oh,
And
yes, sir. Yes, sir.
another,
Thank you for knowing that.
I just wanted to add a few more things because there are
Yes, sir.
some pharmaceutical bioanalysis colleagues also there. This is what
Yes, sir.
need to
Yes,
be
sir.
done. You cannot
Yes.
afford to just do some extract in some animal model and all. No, that
That is the reason, sir. I
the
am
old
in.
way of doing things. So
Yeah, sir.
we have to utilize the modern
Technologies.
bioanalytical technologies that are there.
Yes.
And nowadays, like HPLC and UPLCs and
LCMS.
uh,
Everywhere there, sir.
LCMS are becoming order of the day. So
Sir.
that is very important.
That
And
that
another
that is
thing
that
is,
made me to so much of interest to share my knowledge with the fellow colleagues, sir.
good. And another thing is, the some of the techniques, especially ultracentrifugation, microdialysis, all these things which you have explained in the context of natural product natural product extracts and active ingredients. Actually, these techniques, especially ultracentrifugation, microdialysis, are heavily used in the various DMPK assays.
DMP cases.
Especially in yesterday, I was mentioning about plasma protein binding,
Yes, sir.
finding out a bound versus unbound fraction of a particular drug and all those things. So
Yes,
these
yes.
technologies are actually used in various DMPK assays and DMPK and bioanalytical with the help of a HPLC, with the help of LCMS. People directly go into and understand what particular, based on the mass, they will try to identify and configure the structure and all those things. So
That's what.
some of these technologies are used across all, across the total drug discovery, and especially related to phenotypic aspect. That is the
Yes,
reason
sir.
why, as you rightly mentioned, Chinese are utilizing these technologies very well.
Left and right they are using, sir. Left and right they are using.
They are very well using these technologies, and I don't know. I real
Sir, if you believe me or not, whichever the research papers which I started preparing the slides for me, all the research papers are from the Chinese people only.
yeah. That is there. I think I really don't understand sometimes what
Why
really
we people are
comes.
not
Like
using? That is the
most
thing which I am
more
worrying.
nowadays.
Our fellow colleagues, our
Yeah.
own teachers, they should learn and they should practice this. Then definitely their students will do work. That is the reason I am insisting on to this to do this
Yeah,
kind of work.
it is very important to inculcate research oriented mindset in students. That's very
That's what,
important,
sir.
and
That is the reason I am.
that is the key. And nowadays, even in academic academic colleges and all, also people are having HPLCs and LCMS and all these things.
Yes.
And
That's
you
what,
know, even if we don't have to have all equipment. Even if okay,
no need to.
you have isolated something and you wanted to know what is the you know, like what is the protein NMR of the particular compound or something, you can send a sample to some lab where NMR
Various contract research labs are there, sir.
and one can do the protein NMR, and they will actually, the protein NMR expert can actually decipher and try to give possible plausible structure of the molecule
Yes.
also. So these
Yes, sir.
kind of things, you know, I hope in Indian academic also people will extensively use these kind of techniques, especially because why I am saying is the research that is being done in an industry is totally different, but in various academic institutions in India, still a lot of people try to study old age Ayurvedic products
Old
and
methods
herbal
only.
extracts and all. But that is fine.
That's what.
Doing and evaluating Ayurvedic extracts and herbal extracts is very much fine. But only thing is they should start using this kind of advanced technologies
Yes, sir.
to bring more value to the work they are doing, so that we can publish in more international journals and all that kind of things.
That's what, sir. That's what. No, why we cannot get one Nobel Award by using plant active components? See, 2015 artemisinin is the plant active components.
Yeah.
In that way, I am thinking, and I have shared this my knowledge with my
Good
fellow.
that you have brought you presented this as a complementation, because it is not possible for one particular presenter to present everything.
Yes, sir.
So
Yes, sir.
it's
Yes,
good to see that you have what I have presented yesterday totally industry driven
yes.
drug discovery. You presented the complementation for that phenotypic discovery and how various technologies can be used in phenotypic discovery.
Yes, sir.
A good effort.
Thank you for your comment. Yes, sir. Yes, sir.
Thank you, sir. Thank you, Srinivas, sir, for adding the points. Thank you so much, sir. Now, if anyone of the participants still, if they wants to interact with the speaker, the session is open for the discussion. Okay, as we are running out of the time, we need to close the session, sir. Now I request Dr. M. Srinivasu, sir, Director R&D, Shantaram College Pharmacy, to kindly propose a vote of thanks. Before that, with the permission of the principal and organizing committee, we would like to felicitate our today's keynote speaker, Thippeswamy, sir, virtually. Dear sir, kindly accept our, sir, if you don't mind, can you please stop the, yeah, thank you, sir. We would like to felicitate you virtually.
Thank you. Thank you so much. Thank you. Thank you so much, sir. Thank you. Thank you so much.
Thank you for your valuable time also, sir. Thanks a lot.
We will be in touch.
Sure, sir.
We will do a lot. Okay.
So that's the thing. Thank you so much, sir. Now I request Srinivasu, sir, to kindly propose a formal vote of thanks, sir.
Thank you, Ishak. It's my immense pleasure for taking this opportunity to thank today's technical expert and keynote speaker, Thippeswamy, sir. And I foremost, I would like to thank our chairman, sir, Dr. M. Shantiramudu, sir, who is all given us this opportunity and explore the new possibilities, different techniques for the budding pharmacist and international wide, and emphasizing this to be useful for the many of the people in the pharma field. And I thank our Dr. Ashok Kumar sir, Director of Admin, Shantiram and Arjum Group of Institutions, who is cooperating and supporting us in all the ways. Whenever we are taking the proposal, without hesitating for the cost and time, is giving permission to act, and hence we are able to do this kind of improvement programs, which is actually bridging the gap between the academics and the professional requirements for the student community as well as the faculty also. And I thank our principal sir who has given me this opportunity and who is a driving force for conducting all such activities and making all these events a grand success. I thank especially Thippeswamy sir, who has accepted our invitation with a very short notice and prepared an excellent presentation, which is a combination of medicinal chemistry, analytical chemistry, and pharmacology, because these three fields are very important in drug discovery, where medicinal chemistry, combinatorial chemistry methods are involved, and the bioanalytical methods involved from the chemistry part, and in vitro and in vivo screening models involved from the pharmacology department, which actually requires for development of a new drug entities. So this also made us to think, as you know, sir, to come out of the box from the classical methods of research to the towards new methods. Certainly, this is a big turn towards we all from hereafter work towards that, and we form different research teams, and we'll see the possibilities of presenting good papers. As you know, the papers from for the essay, science edit index, and all, it is becoming very difficult until otherwise we are opting for this novel methodology techniques. So in such a way, you are really shown us taking the torch in your hand in the dark space of in front of us. Thank you so much, sir. And
Thank you,
I
sir.
thank all the principals who have actually encouraged their faculties and students to attend this program and making this all the new methodologies, excellent resource persons, the four resource persons we have found. All these lectures are actually brought to the student community to enrich their young minds and making their academics and research a new beginning. And I thank personally our experts like our Ishak sir, who is actually the technical expert behind all the success of each and every program. I know the technical difficulties they are going without taking any problem, without hesitating at any time. He is the person doing all the technical work behind the screen and also on the screen. So thank you very much, Mr. Dr. Ishak, for your kind help and cooperation. I thank our
Thanks.
team. These are the four persons actually. Ravi Kumar sir, Professor Ravi Kumar, and Dr. Shivaya and Ishak. These three plays a crucial role for successfully conducting, presenting, and linking, making this event a grand success. Though I'm in the front line, because without these three persons, we are not able to make a grand success. And though we have various participants from across the world, from Jamia Hamdard University from Delhi, and the different states of the India, and a few from Indonesia also. We thank all the pharma colleagues for giving your supporting hand for us and making this opportunity useful to you. This will make us more motivating us to do this kind of programs more and more. With the resource persons available, with their support, we will extend this kind of programs for the betterment of the pharmacy profession in India. Thank you, sir. And I thank our board principal, sir, especially, who is actually guiding us and giving all liberty to conduct this program in a well organized manner. And I personally thank all the students, staff in charges, and technical staff, non-technical staff, and all other unmentioned. If I lost anybody, for successfully completing this session. Though today's a Sunday, many of the people are really sitting in front of personal, investing their personal time and making this event. My humble thanks to them, and bow my head in front of you for supporting this farmer field to this extent. Thippeswamy sir, my special thanks to you for accepting your invitation very short notice
Thank
and
you very much, sir.
preparing. Even I have not given you even 1 week time also for preparing the slides. You came very nicely and presented a beautiful seminar. And the rest of the 3 seminars also were excellent, which are very informative, easily digestible manner. With this seminar, actually, for our students, our staff members, and the rest of the community members, it's going to be a eye opener in the field of research for the better publications. However, we are doing research, but to get published in the well-run, reputed journals which are there in the science cited index, it could be a certainly useful method, sir. Thank you, sir. Thank you all. And thanks, thanks for all the patiently listening, all the participants, and thank you one and all. Thanks for giving this opportunity, Mr. Shaq, and for our principal, Dr. Mohsen Chetty, sir. Thank you.
Thank you, sir. Thanks a lot, sir. So this is the session. Thank you so much for your kind support and kind love. One more thing I would like to happily share that all these videos, all these webinars, what we are having, we are going to upload in our YouTube channel very soon. So if any one of you want to have additional information, you can subscribe. You can visit our YouTube channel, subscribe and share and like the videos. So kindly support us. Thank you so much. Signing it off. Jai Hind.
Thank you. Thank you.
Thank you. Thank you. Thank you.
Jai Hind. Thank you, sir. Bye.